Everything alive runs on the same four-letter code: A, T, G, and C. Just four letters, but spell out 3 billion of them in the right order, and you get a human being. Change a single letter, and you can change a life. This is how that works. Those letters are molecules called bases, strung along a long backbone, and they pair by a strict rule: A always with T, G always with C. Two matching strands twist around each other into the shape you know, the double helix. That pairing rule is the whole trick. It is how DNA copies itself, each strand a template for the other. So, what is a gene? A gene is just one stretch of that code, a single instruction, usually the recipe for one protein. You have about 20,000 of them, spaced along those 3 billion letters, packed into nearly every cell. Most of your DNA is not genes at all. The genes are the stretches that actually get read. Here is the part that turns code into you. A gene never builds a protein directly. First, it is copied into a working message. That is RNA. Then, a molecular machine reads that message 3 letters at a time. Each 3-letter word, a codon, calls for one amino acid, and the amino acids link into a chain. That chain folds into a protein, a tiny machine that does an actual job. DNA to RNA to protein. Biologists call it the Central Dogma. Now you can see why one letter matters. Swap a single base and the codon can change, and a different amino acid goes into the chain. One wrong building block can bend a protein's shape or stop it working. In sickle cell disease, exactly one such swap turns round red blood cells into stiff crescents. One letter, one protein, one life. That is the story every episode of this channel tells. For most of history, all of this was invisible. Then, in 1977, Frederick Sanger found a way to read the letters in order, to actually spell out a gene. That one advance is why we can now find the single base that is out of place, and, increasingly, fix it. Reading the code changed everything. It is how we trace a disease back to its source, how we read ancestry, how modern medicines get designed. Every gene has a story, a discovery, a mechanism, a consequence. DNA to gene, to message, to protein, to you. 4 letters, read in 3s, written 3 billion times. Now you can read them too. The Gene Channel.